Hi guys, today we're going to look at 2.2, cell size and compartmentalization. Okay, so cells are typically microscopic. That is, they are not really visible to the naked eye. And so when we talk about that kind of scale, we're usually talking about micrometers. A micrometer, represented by this particular letter here, is 1/1,000,000th of a meter, or 1/1000th of a millimeter. Bacteria are typically 0.1 to 1.5. Plant cells are about 10 to 100 micrometers, which is 0.1 millimeter, and animal cells are 8 to 60 micrometers. And here is a bit of a scale for you to kind of get an idea. Every time you move up, you move by a factor of 10, so it's not a linear scale, rather it's more of a logarithmic scale. And you can see there, things like lipids and proteins are really tiny. And then things like mitochondria and bacteria sit about the 1 micrometer. Plants and animal cells sit somewhere around here. And then once you kind of move past that, you begin to be able to see things with the naked eye. A frog egg, for example, is 1 millimeter, and then a human is ranging in the scale of meters. Anything kind of beyond a millimeter probably needs to be looked at under a microscope, and then anything kind of beyond 10 micrometers probably needs to be looked at using what we call an electron microscope, which works in a very different way from a regular light microscope. All right, now to understand why cells are so small, you need to understand a principle called the surface area to volume ratio, right? It is the ratio of an object's surface area to its volume. If you want to consider how cells are going to survive, all cells must be able to do a few things. They must be able to exchange nutrients and exchange wastes with their environment. You want to collect the good stuff and get rid of the bad stuff, right? And on top of that, the organelles inside the cells must exchange materials with the cytosol. So, if you were to imagine the organelles kind of functioning and relying on each other, they need to be able to exchange materials with one another inside that particular cytosol, okay? As a cell grows larger, however, right, its volume and its surface area are going to increase, but they actually increase at different rates. The volume usually increases faster than the surface area, right? That means that it's going to slow down the cell's ability to exchange materials that are going in and out of the cell, right? Because the volume is growing, but the surface area is not growing enough to match all of that exchange needs, because cells exchange things through their surface area. Beyond a given size, the cell actually can't sustain itself, and it either has to divide into two or it will die, right? So if you were to think about the cell, kind of this is kind of your shape of the cell, and all of that material that goes in and out of the cell, that needs to move somewhere, and the bigger the cell gets, the more distance it needs to move, right? Here is another diagram showing the change in ratio, which you can see as it goes bigger, the surface area to volume ratio actually goes down. So here's a small block, right? And if it is 6 centimeters in surface area, but only 1 centimeter cubed in volume, the ratio is 6 to 1. If you double the length of the cell, and it is now 2 centimeters in width, length, and height, it's going to have a surface area of 24 centimeters squared, but a volume of 8 centimeters cubed. And if you take the ratio, it is now a 3 to 1 ratio. And so as cells grow larger, their surface area to volume ratio goes smaller. Okay, here's the same thing represented. So if you were to think about it, for a cell, the advantage is actually to stay small, because that gives you a greater surface area to volume ratio when you stay small, right? So if you have, let's say, for example, 2 cell volumes, both of which make up 125 units, right? If you chop this particular block up into heaps and heaps of 5 by 1s and so forth, right, you're going to maintain that particular cell ratio, given that you don't block them in with each other, as 6 to 1, right? But you keep it as one large unit, right? It's going to be very difficult for the cell to exchange materials from the center to the side, and so that slows down the cell's ability, right? So that's kind of giving a kind of basic principle on how surface area to volume ratio works. So here's an example of a cell kind of living its sweet life as a single-celled organism, being so small, able to exchange materials with its environment, like, for example, just grabbing food straight from its environment without having to worry too much about getting it to all of its cells. And then as you go larger, you can see there, this is kind of your first multicellular multicellular kind of organism with clusters of cell working together in this kind of gel matrix kind of material, but starting to maintain their surface area to volume ratio. Okay, increasing your surface area to volume ratio, there are basically 3 ways that you can increase your surface area to volume ratio, right? Number 1, you can divide into many cells, right? So it's going to keep the materials in the cytoplasm pretty close to the plasma membrane boundary for exchange. In other words, whatever's inside the cell is pretty close to whatever's outside the cell, which makes exchange of materials easy. You can flatten yourself, right? Retains your total volume, but your total surface area is going to be very wide and stretched out, which allows the material a shorter distance to get to the outside world. This is a flat worm, being a very flat organism, allowing itself to just absorb oxygen straight from its environment pretty quickly. The third way you can do it is you can actually make extensions, right? The extensions will thin out the volume but increase the surface area for efficiency. So for example, plant roots form these extensions, which then allow the plant to maintain a low and as much of a volume as possible, but increasing its surface area to absorb things like water and nutrients and so forth, okay? Now, when we think about cells, we have to also think about compartmentalization, right? So in the previous chapter, we talked about one of the key characteristics of eukaryotic cells is that they contain membrane-bound organelles, right? Now, this particular trait is called compartmentalization, that kind of compartmentalizing yourself into different parts. Here is kind of a diagram and a electron microscope kind of micrograph of a animal cell and a plant cell, and you can see, if you look very carefully, there are lots and lots of little structures inside of our cell, right? So there is lots and lots of little green structures that are here, lots and lots of nucleuses and nuclei, and lots of these little kind of plots here in the plants, and you got these ones here in the animals. We will go through into each of them in detail, but compartmentalization is what we're looking at here, right? The idea that the cell has specialized membrane-bound compartments that are called organelles, right? By compartmentalizing itself, it allows each organelle to have a different function, and then the different compartments to have different internal environments. Like, for example, the stuff that's in here is going to be different from the stuff that's in here, for example, right? And this one might have enzymes and this one might not, for example, right? And by doing that, the cell becomes less vulnerable to changes in its particular environment, right? Now, in the next chapter, we will look at what these organelles actually are and how they actually work together to make the cell more efficient, okay? But that's it for this particular chapter, and I hope you guys enjoyed it, and I'll see you in the next video.